So I took a pause there because I needed to allow Google Chrome to record my screen because that is going to be the format of today's lesson. I am going to do a screen recording of a document that I prepared for the lesson. And I'm also going to add notes in real time during this live stream that go deeper onto the topics that are outlined in the document that I'm going to share. So this is going to be in Google Docs. I'm going to put the link in the chat, and that link is going to be public. So literally anyone who's viewing this now or later, once it is published on my YouTube channel, will be able to go and see the document as it is at the end of today's live stream. So let me introduce myself. My name is Aiden Malanaphy. I'm a private MCAT tutor in New York City and online. I'm usually in a tutoring session with someone, either on this laptop, online, or in person at a library or anywhere generally in the New York City metro area that a student needs me. I work mostly in Brooklyn right now because I went to Brooklyn College to take my science classes before I applied to medical school. So just a little bit of background on me. I'm going to be starting my MD this August. August 4th is when my orientation starts at the Albert Einstein College of Medicine in the Bronx. So I was accepted to medical school earlier this year. And I've been tutoring full time for a couple of months now and trying to grow the business full time. And this is part of me growing my business and trying to reach a larger audience. I used to tutor chemistry at Brooklyn College, and I found that there was a lot of interest in live stream lessons that I did at Brooklyn College and had live streamed on my YouTube channel. And you can go and view those if you're a chemistry student. Okay. So I'm going to open this document. It's going to take me a minute because I need to change the share settings on it. And this is going to be like the guide to today's lesson. It is going to serve as a guide both today and in the future for anyone who's viewing this video posted on my site, my YouTube channel rather. Okay. So I'm going to go in. Okay, this is going to take me a minute. I'm sharing a document on Google Docs. So I'm changing this to anyone with the link. It's going to become public. Done. I'm going to copy the link. And here it is posted in the chat. So that's a link to a Google Doc. If you click it, you're going to be taken to Enzyme Kinetics Aiden MCAT live stream. And that is the name of the document that I'm going to be taking notes on today as I go through today's topics. Okay. So now, without further ado, I'm going to jump onto that document and I'm going to start working on it. And as I work on it, I'm going to look into the camera and explain what I'm typing and type the things that I'm explaining. All right, so I'm going to now share my screen. It's going to show this. Okay. So, okay, there we go. Perfect. This is exactly how I wanted it. So I haven't done a YouTube live stream before in this format. I've only live streamed live videos, but this is the document. So enzyme kinetics is a very important topic for the medical college admissions test. I guarantee that if you're taking the test anytime in the next few years, enzyme kinetics is a subject that will be tested in some way or another on test day. I want to preface the lesson by saying that you don't need to be an expert in enzyme kinetics in order to know the enzyme kinetics you need to know for the MCAT. MCAT test takers are not taking the test to get a PhD in biochemistry or to pursue a biochemistry topic such as this one or physical chemistry topic such as this one in greater detail. We're being tested for our potential to be good medical students and doctors. And that is much more about making quick determinations and initiating an effective action based off of that quick determination, just like when you're treating a patient. So this is the date, and that's, ideally when we would have started, but we're starting about 10 minutes late. And link to the video recording. I'm going to put that later on once we're done with tonight and we're all wrapped up. And then these are sources that I got all of the information from today from. So I'm an authority on absolutely none of these topics. I have a bachelor's of arts from Wesleyan University. So I'm an authority on Lin-Manuel Miranda plays, and really not even that. So, this is where I got this information from, not me. None of this is stuff that I came up with. This is all pretty foundational science, if not outdated at this point. So here are my sources. The first is this biochemistry textbook, Concepts and Connections, by Matthews, Appling, and Anthony Cahill, specifically chapter 8, Enzymes as Biological Catalysts, because that's the topic of today's lecture, enzymes as biological catalysts. The second source is this open source textbook called Chemistry, and it's the second edition by this Rice University initiative called OpenStax. And their license says, except otherwise noted, textbooks on the site are licensed under Creative Commons Attribution 4.0 International License. Basically, anyone could use them. That's why I'm using this. It's a free textbook. You can click that link and have the whole book at your disposal, which is pretty awesome. Source number 3. This is from the Association of American Medical Colleges, which I would take as gospel because that is the entity that produces the MCAT. They're going to write your test. So I would pay attention to everything they say. I take a lot of information from the What's on the MCAT exam PDF that they published in 2020. And that's copyrighted. So we're going to look at it. We're going to actually use some content from it farther down. Then I'm using this Miles Down MCAT review sheet, which is a study aid that was produced by this entity or group called Miles Down. Honestly, I'm not sure where that comes from, but it was very helpful to me when I was reviewing for the MCAT. And so I'm using it now in this live stream as sort of the most pedestrian or accessible way to explain any of the topics we're going to get into today. Okay, so from here on out, I'm going to be using the AAMC as a guideline and adding in all of my own, thoughts on what the most important points are in response to each point that the is difficult to understand, but the AAMC gives a content outline for what they want you to study for the MCAT. And today, I want to just stick to that outline and just for about an hour, knock through all of the concepts under the heading of enzyme kinetics. So we're going to stick to what the AAMC wants us to know, and we're going to add all of the content on top of it. So the AAMC has these things called content categories for what they want you to know for the MCAT. This content category that we are diving into today is called the structure and function of proteins and their constituent amino acids. The structure and function of proteins and their constituent amino acids. Enzyme kinetics falls under that category, but it is not the only topic that is covered in content category 1A. Even more broad than content category 1A is the foundational concept that encapsulates everything we're going to be discussing today. And that is the one about biomolecules. And I'm going to read it because it's straight from the AAMC. And I think basically everything that they say deserves your attention if you're going to take this test. Biomolecules have unique properties that determine how they contribute to the structure and function of cells and how they participate in the processes necessary to maintain life. So biomolecules include things like enzymes. They also include things like carbs, fats. It's a broad category. That's why I put it at the top here, because it's sort of the umbrella. Content category 1A. And then continuing to content category 1A, we're going to read this description. It is long-winded. It's very verbose, but that's how the MCAT delivers information to you. And so it's good to get used to reading things like that. A long-winded paragraph that has the information you need, and you just need to figure out how to find it. It gets easier as you study more, which hopefully we're going to be helping out with a little bit today. Okay, so category 1A focuses on the structural and functional complexity of proteins, which is derived from their component amino acids, the sequence in which the amino acids are covalently bonded, and the three-dimensional structures the proteins adopt in an aqueous environment. I'm just going to explain a couple of parts of this that might seem a little random if you might be like earlier in your prep, or if you just don't know. It's never a bad time to ask a basic question because the MCAT asks basic questions. So proteins are comprised of amino acids. So that's why the sentence includes this part about the fact that the complexity of proteins is derived from their component amino acids. Then, with those amino acids, they could form many different combinations, innumerable, and the sequence in which those amino acids are bonded together, and we know that the chemical bonds that they're forming with each other are covalent chemical bonds, not ionic chemical bonds. So covalent, sharing electrons equally, quote unquote. And the three-dimensional structures, okay, so proteins are three-dimensional. Good evidence for that is the human body. It's not flat like a pancake, and that's because we have, well, the skeletal structure, but our tissue is formed of proteins. So that the three-dimensionality of that goes all the way down to the molecular level. So the three-dimensional structures, the proteins adopt in an aqueous environment. Aqueous just means watery. And something that I have been hearing since I was a kid is that humans are mostly comprised of water. And so you'll be seeing that really whenever we're talking about something inside of the human body on the MCAT, you can assume that it might as well be happening in a beaker with water and a bunch of other stuff, but water primarily, which isn't really the case in real life, but we're just going to call it that because it really does apply in the vast majority of scenarios. Okay. So now let's get into these headings. So enzyme structure and function. That is the heading. And then the function of enzymes in catalyzing biological reactions. Okay, so this is where I'm going to pull up the chapter 8 of that textbook because we need to define what an enzyme is. The quick and dirty definition that comes to mind for me is that an enzyme, so I'm just figuring out how to type properly with this, is a protein that catalyzes a chemical reaction. It's that simple. So this is going to bring us into the next part of the definition. I'm going to pull up the textbook. But this is the basic definition that seems very simple, and it is very simple. But the parts of it become very important when you're studying for the MCAT and when you're taking the MCAT, because they like to really just drill down into like what makes the most basic parts of the foundational sciences, like what makes them what they are. What are the most basic definitions is really what they're asking a lot of the time. So it just becomes important to recognize those, and that's why I'm including them in a lesson like this, because they're utterly relevant. Okay. So we've already established that an enzyme is a protein. We also talked about how a protein is just a long string of amino acids that has a three-dimensional structure. That's so far what we know about enzymes. And then we have this thing about them catalyzing a chemical reaction. So this is what's defining an enzyme for us. not every protein is an enzyme, but every enzyme is a protein. Well, then what makes these things, these enzymes, special? It's that they're catalyzing a chemical reaction. But what is a catalyst? And it might sound like a basic question, but you may get that basic question. Here is that textbook that I earlier, chapter 8. Their terms, they're in general terms. This is their definition for a catalyst. In general terms, a catalyst is a substance that increases the rate or velocity of a chemical reaction without itself being changed in the overall process. So that's what you're going to find in textbooks as the definition of a catalyst. It doesn't always have to be an enzyme. It could be something else. They're just saying here that it's a substance. Um, so it increases the rate or velocity of a chemical reaction, and it is not itself changed in the overall process. So overall process, this means that from the very beginning of the reaction to the end of the reaction, the catalyst is the same at the beginning and the end. In the middle, the catalyst might change its structure, but that doesn't mean that it ends up changed after all is said and done in the reaction. A good way to recognize a catalyst when you're looking at an equation for a chemical reaction is to see that it's the same molecule before it goes into the reaction and after it goes into the reaction. And so enzymes are going to be the same story because they're catalysts. So this is an important concept that the textbook calls out here, but then I'm also going to include here. So a catalyst a catalyst speeds up a chemical reaction and does not change throughout the overall process. It does change in the middle, but it doesn't change at the beginning and the end. And I would say, this is Aiden's definition now, I would say comes out of the reaction unchanged. So it speeds up a chemical reaction and comes out of the reaction unchanged. So here are the buzzwords: enzyme, catalyzes, catalyst. And we're going very basic because this is what the MCAT is going to test. You need to drill the basic definitions, in my experience, in order to do well. Oh, I should have here. Sorry, I've been over here. So I typed out just now: a catalyst speeds up a chemical reaction and comes out of the reaction unchanged, unlike the reactants. So it being a chemical reaction, the reactants change, the catalyst does not change. And then another thing that the textbook noted is that catalysts only speed up a reaction. They do not make it more or less thermodynamically favorable. This is a very important sentence, really, to know for the MCAT. They make, and I'm going to add this other sentence that maybe isn't often the way that people put it. They make reactions more kinetically favorable, i.e. faster. Okay. So enzymes make reactions faster. They do not make them more thermodynamically favorable. And a good way to visualize this is by looking at the energy diagram of a reaction. So I'm going to share my tab, share the textbook again. This is a really scary diagram, but the MCAT will throw you scary diagrams, and it'll demand that you sort of see the simple diagram within the scary one, and it gets easier the more that you sort of just do this and recognize that what they're really showing you is not that complicated. It just looks dressed up and like fancy. But again, we're not trying to get PhDs in biochemistry here. We're trying to get into the hospital and treat patients. So anyway, this is the Gibbs free energy. It's a thermodynamics number. The Gibbs free energy before the reaction and the Gibbs free energy after the reaction. Usually, you need to have the Gibbs free energy be lower at the end than at the beginning, because nature tends towards lower energy. And a different way that people usually say that is it tends towards more stability. So lower energy is more stability. And so you find more stability in a lower Gibbs free energy than a higher Gibbs free energy. And I'm saying Gibbs with a G, and that's where the G comes from. So higher G over here, lower G over here. The catalyst is one thing here, and it's that exact same thing here. But in the middle is where it's doing its work. So it's another good way to remember the fact that a catalyst does not change the beginning and end energies of a reaction, but it does change the energy in the middle of the reaction. And that is called the activation energy. So it looks like a mountain, and this is the peak. And it is called the activated complex, or there's a bunch of different words for it. But what it basically is the way that the molecules are shaped in the middle of the chemical reaction such that they are at their highest energy state, i.e., they're least stable. So at the highest energy state, the reaction either proceeds this way or this way. Basically, all reactions are reversible. And it's going to proceed this way because of the Gibbs free energy and the fact that it's favorable, right? More stable on this side than on this side. But basically, we're at the top of a hill here. And what an enzyme or any catalyst does is it just makes that hill a little bit less steep. It doesn't change where you started, it doesn't change where you ended, but it makes that hill a little bit less steep. And it means that you'll get over the hill a lot faster. And by a lot faster, I mean like way faster. The time scales across these catalysts work compared to what the speed of reaction would otherwise be is mind-boggling. So this is a really a great way to show it. So these are enzyme rate constants. We're going to talk about that later. But basically, just think of it as like how fast the enzyme works. So for a catalyzed reaction, the reactions are happening like below 1 second in terms of speed. So they're happening instantaneously. And then for these uncatalyzed reactions, it could take up to, for this one, up to 4 billion years. So it's sort of like an exponential kind of time difference. And what I take from that is that this is how our body is able to do chemistry, because we don't have time to wait for these reactions, these chemical reactions to happen without catalysts. And that's why we have catalysts. And then I'm just going to recap what we have already talked about. So under this AAMC heading in content category 1A, function of enzymes in catalyzing biological reactions, we've learned that an enzyme is a protein that catalyzes a chemical reaction. We've learned that a catalyst in general speeds up a chemical reaction, and it comes out of the reaction unchanged, goes in one way, it comes out the same way. And we also learned that a catalyst only speeds up a reaction, i.e., it is making the reaction more kinetically favorable. It is not making the reaction overall more thermodynamically favorable. The thing that what makes a reaction more thermodynamically favorable is the beginning energy and the end energy. Enzymes are chiefly concerned with what is happening middle. They're not about the beginning and the end. They're all about what's happening in the middle. because the middle is what takes up the most time. So they speed it up exponentially, as I was explaining earlier, and that's by working in the middle. Okay, so we've established that enzymes are acting within the context of a chemical reaction. And stepping outside of biochemistry for a second, we learned countless kinds of chemical reactions in general chemistry and even more in organic chemistry. So given that there are many kinds of chemical reactions and a few that are really important in a biological context, there are also many kinds of enzymes and a few that are very important in biological contexts. And those are the ones that we're going to be most concerned about for the MCAT, because we're relating what we're learning to treating human beings. Okay, so I'm going to just go and find now I'm going to use this source, the Miles Down review sheet. This was like my Bible when I was studying for the MCAT. Enzyme Kinetics. Okay. So this is a big and very dense document. I was always a little bit scared by its density, but the purpose of it being so dense is to fit the entire document into 90 pages. So just keep that in mind. This is how all information is going to be presented to you on the MCAT. So what we want to find are the different kinds of enzymes. Okay, here we go. So enzymes as biological catalysts. So they actually use the same heading as, or I guess not, but function of enzymes in catalyzing biological reactions. It's the same heading as the AAMC. That's why I feel pretty comfortable using Miles Down. And if you go to any of these more reputable and like institutional sanctioned sources, they'll corroborate all the information that I found in Miles Down 9 times out of 10. Some things they get wrong, but I'll try to find that for you. Okay, so we already talked about this. Biological catalysts are unchanged by the reactions that they catalyze and are reusable. Enzymes do not alter the delta G, which is what we talked about, Gibbs free energy, or the delta H, which is the enthalpy of a reaction, or the heat of the reaction, nor the final equilibrium position. Let's not worry about that for now. They only change the rate of reaction by altering the mechanism. So before I said that the catalyst is chiefly concerned with the middle of a reaction. Said the middle of the reaction. So in organic chemistry, we learned this better than in any other class, is that what's happening in the middle of the reaction is crucial. It's the whole reaction. And that's what the enzyme is focused on. It's that middle part. And what's happening in the middle part? It's the mechanism. So we learned a lot of mechanisms in organic chemistry, and that becomes very important when you're studying enzymes at a deeper level. Thankfully for the MCAT, the mechanism of enzyme catalysis is not deeply tested. It's really very surface level. You could go way deeper into how an enzyme catalyzes a reaction than they're going to test on the MCAT. But you do need to know that generally an enzyme is doing its thing by changing the mechanism by which a reaction happens. They both the forward and reverse reaction. So I explained earlier how essentially every chemical reaction is reversible. It's just that nature, via the Gibbs free energy and the heat of the reaction, really strongly favors one side. So a good example of this is the conversion of glucose to pyruvate and a bunch of other energy molecules. The process by which that happens, which is this whole thing called glycolysis, and we learn it in high school and then college and then in medical school again for the boards. It could happen in reverse, and all of the catalysts that are working along that pathway, even the non-reversible steps, hypothetically could happen in reverse. The important point here is that the catalyst will catalyze both the forward and reverse reactions because it's altering the mechanism in either direction. But just because it makes the reaction faster does not mean that it can choose which side the reaction lands upon. The reaction is going to continue to land upon the product side, um, because that's determined by thermodynamics, not kinetics. The speed at which it lands on either side is determined by kinetics. Okay. I would skip this. Okay. So here we get into a bunch of aces. So I'm going to put down here that enzyme types are generally, they end in ace, or enzyme names rather. So aconitase is one example. That's an enzyme that's involved in the Krebs cycle, I think. We're not going to get into so let's go with our first one, which is an oxioreductase. I like how they start with that. I like how Miles Down starts with oxioreductases, because the thing that they do is in the name. They're an enzyme that catalyzes an oxidation reduction reaction. And I'm just going to put in here oil rig. When it comes to oxidation and reduction on the MCAT, every time that I would see it on my test or in practice tests, I would write oil rig, which stands for oxidation is loss, reduction is gain of electrons. So an oxioreductase enzyme is speeding up or catalyzing a chemical reaction that is classified as either an oxidation or a reduction. And every redox reaction, which is sort of like our abbreviated term for these kinds of reactions, a redox reaction or reduction oxidation. Every redox reaction is both an oxidation and a reduction, because it takes two chemical species in order to make the reaction happen. So to one of them, it's an oxidation, to the other, it's a reduction, and thus we have a redox reaction, and the enzymes that catalyze that kind of reaction are oxioreductases. Okay. Another really important kind of enzyme is a transferase. So a transferase also does what's in the name. It transfers alkyl group from one reactant to another. A good example of a transferase is a kinase. Which add phosphate groups. And a phosphate is a chemical group that is transferred from one molecule to another. The molecule that it's usually transferred from is this one called ATP, adenosine triphosphate. So it loses one of those phosphate groups, and the enzyme that catalyzes that process, i.e. speeds it up, is called a kinase. And the general category that the kinases fall under are the transferases. Okay. So after this, we have hydrolase. Hydro water. So the hydrolase is going to use water to break up a chemical bond. And when I say the hydrolase is doing this, I don't mean that they're actually causing the reaction to happen. I would say that that's the domain of thermodynamics. They're speeding up a process that is already thermodynamically favorable. They're making it more likely to happen on a biological timescale. So it uses water to break up a chemical bond. What I mean is that it's speeding up the process by which water is used to break a chemical bond, because that's the nature of an enzyme. Okay. And then we have isomerases. A molecule to an isomer of itself. So an isomer, iso is like a prefix that means the same. Same. So iso. And then mer, we're not going to do that. But Isomerase changes a molecule to an isomer of itself. The isomer of a molecule is a substance that has the same chemical formula if you wrote out the atoms and the number of each atoms, but it has a different arrangement of those atoms in three-dimensional space. So an example you may remember if you've taken organic chemistry is enantiomers. Um, enantiomers. I'm just going to call that Aiden's example of isomers. Enantiomers are mirror images of each other, and that means that the arrangement of their atoms in three-dimensional space is different. Hypothetically, I guess an enzyme could catalyze that transformation, but what we're usually talking about is um, changing a molecule like a metabolic intermediate into an isomer of itself by just rearranging the chemical groups on it. Because often our body wants things just in a different sort of form than it comes in from nature. So isomers are just, iterations of the same molecule in different arrangements. And isomerases speed up those processes. A ligase is an enzyme that joins biomolecules together. So when I think ligase, I think ligand. So a ligand or a ligand is a group that is added to another group. And the reason that I use that as sort of like my definition, or my way of memorizing this, is that a ligase is adding two groups, which are biomolecules together, to make a larger thing, which often in the body you need to do. I mean, we have proteins comprising, a lot of what you see here, and in order to form those massive biomolecules that we have supporting our bodies, you need enzymes that are going to speed up the process of sort of combining things together, almost like a railroad track, where you have these big pieces, but they need to be put together in order to make these huge railways that we have. That's maybe like an analogy for that. Okay, now we have lipase. Lipase. So it's the definition can be found in the name. A lipid is a fat molecule, and a lipase is an enzyme that is going to speed up the hydrolysis of fats. So speeds up the hydrolysis of fats. And hydrolysis just means water being used to split up. So lipases speed up the hydrolysis of fats. Kinases are kind of transferase. I'm reading off the miles down MCAT review sheet. And then, okay, lastly, I want to talk about lyase. Lyases are just like hydrolases or oxyreductases, except when they cleave a chemical bond, they don't use water and they don't use a redox reaction. So that's a definitional thing to know for the MCAT. Sometimes the MCAT is testing things that aren't necessarily like real world relevant, but the definition is very important for your test taking knowledge because they're going to test the definition itself. And lyase is a good example of that. Cleaving a bond without hydrolysis, without oxidation reduction. So literally what you need to know is not exactly what it does, but what it doesn't do. And that's a definitional thing. It's sort of like a process of elimination thing. And that is a lot of the MCAT process of elimination. Okay. And then the last one that the last two classes I want to add, under transferase, I want to have kinase. I also want to have phosphatase. Okay, so this is going to be adds a phosphate group, singular. And this one is going to be takes a phosphate group. And the way I like to memorize that is a phosphatase is like phosphatake. So takes a phosphate group. Whereas kinase is going to add it. And there's this other thing called a phosphorylase, which adds a phosphate group, notably glucose. And you'll notice that these are overlapping definitions. The difference between them becomes clear when you answer questions. Okay. And frankly, I don't really know the difference just off the top of my head, but I do know that we use kinase more often to talk about molecules that make things happen in the body by throwing phosphate groups around. Okay. Again, I'm not an authority on this, but I cited the sources above. I would consult those for more full definition. I already talked about this, the reduction of activation energy. And I'm just going to add that this is achieved by changing the mechanism of a reaction, i.e. the stuff that happens in the middle of the reaction diagram. Okay, and by the middle of the reaction diagram, I mean this part, the part where the reaction, so E here stands for enzymes, S stands for substrate, or the molecule upon which the molecule or molecules upon which the enzyme is acting. So E plus S, and then in the middle is ES, and then EP, so enzyme substrate, enzyme product. But what this is the middle of the reaction, which is just the broad strokes thing that you reduce the activation energy, i.e. the peak of energy in a reaction diagram, by changing the mechanism of the reaction, changing what happens in the middle of the reaction. Okay. Then there's this AAMC heading called substrates and enzyme specificity. Okay, so I'm going to refer again to the miles down sheet because it's really useful, and it puts things in just a really straightforward way. So there's two terms that you need to know when it comes to the specificity of enzymes. And one is the active site model, and the other is the induced fit model. So none of these are really the last word on what's happening in the middle of an enzyme reaction, an enzyme-catalyzed reaction, but they're both tested on the MCAT, and so you need to know both of them. That's often the case here. Is like it's not necessarily what's most important scientifically, or what's most relevant, or what's most current. It's what the AAMC wants you to know. So that's what I teach. Okay, so the active site model is saying that enzyme and substrate lock into place, and the reaction is catalyzed like the turning, this is just my words, the turning of a key in a lock. Okay, and the whole point of a key is that it only acts upon one lock, or one kind of lock. So in the active site model, the substrate fits just perfectly into the enzyme. The enzyme is sitting there, and later on you get your product, and it takes a lot less time than it would if the enzyme wasn't sitting there. The induced fit theory is just a more dynamic story of those same events. So you still have the substrate, which is the molecule that the enzyme is going to catalyze the transformation of. And you still have the enzyme, which is the protein catalyst that makes the reaction faster. But in the induced fit model, they both sort of contort themselves to fit properly, rather than one fitting into the other like a lock and key. So the enzyme and substrate both undergo conformational changes to interact fully. changes to interact fully. Both move around. Okay, awesome. So as you could tell, these are pretty absolute theories, or hypotheses rather, and neither of them accounts for all of what's happening in the reaction coordinate when an enzyme is reducing the amount of time that it takes for a chemical reaction to happen. They're both just sort of hypothetical. Okay. Cofactors are non-protein compounds that help an enzyme catalyze a reaction. A really good example of this, and the most common one is metal cations, calcium, Ca2 plus, lithium, La plus, sodium, um, chlorine. I don't know if that's certainly not a cation. So let's just keep these cations here for now. It illustrates the point just as well. A cofactor, I like to think of cofactory, which means not a very hospitable / biological thing, i.e. not a protein. So this is a mnemonic device that I'm using to remember the fact that the word cofactor means non-protein compound that assists, if you will, the enzyme in catalyzing the chemical reaction. Cofactor is like cofactory. Okay, and then coenzyme equals an enzyme, or a protein that helps an enzyme catalyze a reaction. Let me fact check myself on that, because I'm not sure if it's actually just proteins. It might also be. I'm double checking because the miles down review sheet gives a different definition than this. Cofactors are non-protein molecules and coenzymes. Okay, so let's change that definition because I realized that cofactor was correct in that it's non-protein, but coenzyme is going to be an organic molecule. And a really important example of these are important is going to be vitamins. Okay, and that leads nicely into the AAMC's next topic. Important example. Okay. I'm going to put here the fat soluble vitamins. Those are the ones that I remembered. So water is a polar solvent, and fat, aka lipids, are a nonpolar solvent, and vitamins, which are these coenzymes, are either water soluble or fat soluble. So certain vitamins need fat in order to participate in this chemical reaction because they need to be able to like move around in three-dimensional space. Okay, so the fat or lipid soluble vitamins are A, D, E, and K. A deck. don't remember memorizing this. It just sort of stuck in my brain that A, D, E, and K were the ones that are fat soluble. So any other vitamin are water soluble. I think that would be by definition. Let me just double check that in real time. Okay, so yeah, it's really one or the other. Fat soluble, soluble in fats, water soluble, soluble in water, and also maybe other polar solvents. But luckily, we're only dealing with water when it comes to the human body as the primary polar solvent. You wouldn't want it to be something else. Water is pretty amazing. It is a singular molecule, so it's unique. Okay. So we did the fat soluble or the water soluble vitamins by way of doing the fat soluble ones. Okay. So effects of local conditions on enzyme activity. Honestly, I don't know what this is getting at. I think I would say look into this yourself, but also that things like temperature affect enzyme activity. Too hot and or cold and enzymes stop working. So this is what comes to mind because it can be deadly if your body gets too hot, um, or if your beaker that has the enzymes that you don't want to degrade gets too hot. Because when an enzyme degrades, it can no longer catalyze the chemical reaction. It can no longer speed up the chemical reaction that it's there to speed up. So that means that those chemical reactions will take just an essentially infinite amount of time to take place, and they won't take place at all. And you need chemical reactions to live. You need them to be happening on a biological timescale, rather. So that's part of why our body keeps temperature in such tight regulation, because enzymatic function occurs at an ideal average temperature. And outside of those boundaries, they don't function as well. Okay. That's what comes to mind. I think having your cofactors and coenzymes around is also really important. Having enough cofactor. Okay, so an example of that is the cations that we talked about earlier, like lithium or calcium or sodium, or the vitamins. So you see how often there are these like chemistry definitions and then like the biological definitions of the same topic happens often in these like pre-medical science classes. So a cofactor includes really basically we're talking about metal cations. A coenzyme, really we're talking about vitamins mostly, but and also other important biomolecules. But these larger terms reduce down into really more specific things because these are the things we see most often in biology. And that's what's important on the MCAT, what we see most often. And again, I can't stress enough the analogies to work in a hospital or work in a medical setting, because remember, that's the entire point of taking this test. Is that's where you want to end up. That's why you're taking the test. Okay, now we get into actually talking about enzyme kinetics. The control of enzyme activity is a header. And because this seems so sort of intimidating, especially if you're not familiar with these names, I'm just going to read off all of these headings, figure out how to attack the topic, and then we're going to get into it. So, under the banner of kinetics, kinetics, as we said before, is about the mechanism and the speed of chemical reactions. It is not about the thermodynamic variability. You have kinetics and you have thermodynamics, and they're separate. And today we're only talking about the first one. We're talking about the speed, not talking about the thermodynamic favorability. So, under the banner of kinetics, or how fast these enzymes are working, you have general kinetics, the kinetics of their catalysis. You have Michaelis-Menten enzyme kinetics, which I would just say is the math behind enzyme kinetics. And you don't need to know very much of it, but the little you do need to know is crucially important and will be tested on test day. And then cooperativity. Okay. Second, after kinetics, or the speed of these reactions, is feedback regulation. So feedback regulation is going to be one way in which enzymes are controlled in their activity. One way that our bodies control enzyme activity. And then after feedback regulation comes other kinds of regulation, namely inhibition. And the types of inhibition are competitive, non-competitive, and uncompetitive. And then there's mixed, but we're going to talk about these three first. Okay. And then finally, we have regulatory enzymes. So these are enzymes, I guess, whose job it is to regulate the speed of these chemical reactions. These terms are a little bit less what they call high yield on the MCAT, or like commonly tested. So we're going to get to them last, and we're going to cover them briefly, as we sort of have been with all of these topics. This is a light overview and a refresher more than a 0 to 100 lesson. Okay. So control of enzyme activity. We already talked about catalysis. The nature of catalysis is the speeding up of a chemical reaction by changing what's happening during the chemical reaction. It doesn't change the before, it doesn't change the after. It just makes the after a lot more quick to arrive because it's changing what's happening in the middle of the reaction. So that's what I would say general catalysis. They might be getting at something that I'm just not remembering or covering here. And then Michaelis-Menten. Let's just attack that head on. So I'm going to share my miles down review sheet and type this into, okay. So again, this review sheet is really kind of scary looking thing, but like the MCAT itself, you sort of just have to imagine that it's very simple, because underneath all of this, it's simple, and you need to find that simplicity. Okay, this is the Michaelis-Menten equation. So there are so many steps that you have to take in order to get here, like math stuff, but thankfully you don't have to do any of that for the purposes of the MCAT. But what we arrive at is this equation here, which shows, I just want to see what the live stream looks like right now. Okay, so here it's just the middle of my screen here, and yeah, this is like the headline of the lesson today. What we've been doing so far is like building up. This equation is everything, because it is a roadmap to how fast this enzyme catalyzes its reaction under different substrate concentrations. So I'm just going to say again, this equation is showing us, and I'm going to show. So what I did is I took a screenshot of that equation and I put it into here. So the Michaelis-Menten is just the math. You could think M for math of enzyme kinetics. Again, we're going very basic, very like reductive. We're reducing this down to its essentials. And this equation is telling us how fast the enzyme catalyzed reaction is proceeding under different substrate concentrations, and I'm putting that in caps because that is really important, and that is S. So in chemistry, putting things in brackets, it's concentration. So the MCAT could show you a concentration without warning just by putting brackets around something, and it will expect you to know that that just means the concentration of it. And a way to quantify concentration, one of the units is something like molarity or moles per liter. So the units don't matter because they would cancel out. But the S here is substrate concentration. Okay, so we've already defined one of the variables in this big scary equation, but let's tackle the rest. Okay, V naught or VO is the reaction velocity at a given substrate concentration, aka S in brackets. All right, that is V naught. Okay, V naught. The V is for velocity, and the velocity is just the speed of the reaction. How fast is it going? Okay, and then there's V max, and this is the reaction velocity when the substrate concentration, or no, I would say reaction velocity, the highest reaction velocity. And then, so highest reaction velocity, aka the maximum rate at which an enzyme can catalyze the reaction. This is when the enzyme active sites, which we touched on briefly before when we were talking about the active site model. So here they're talking about active site. That's really just like the key, and the substrate or the thing that is being transformed is the active site is the lock and the substrate is the key. And so what Vmax is telling us is that when all of those locks are inhabited by keys, how fast is the reaction proceeding? And that's the fastest it can go. It's like having a car that is firing on all cylinders. So in the context of an enzyme with multiple active sites, the cylinders are going to be the active sites. And when you're firing on all cylinders, all of those active sites are occupied by substrates. So logically, that is going to happen. Vmax is going to happen when there is the highest concentration of substrate, aka S in brackets, surrounding the enzyme. Okay, and then let's define the last term here. It's the last term, and it's the hardest to understand in my experience, and that is Km. Let me type this properly. Okay, Km. Okay, and that is the Michaelis constant. And that means the substrate concentration at which the reaction proceeds at 1/2 of Vmax. Okay, so let's try to figure out a little bit of what that means. First, I'm going to fact check that with our review guide from Miles down here. Km is the S at which an enzyme runs at 1/2 of its Vmax. So yeah, I'm going to share this part. This is what this review guide often does. It uses these really sort of casual terms for these things, fine, but it's why I also linked to those textbooks, so you can go back and sort of see, well, what does the official quote unquote authority on this subject say? Because it would use a different word. But anyway, digression. Km is the Michaelis constant. The Michaelis constant is the substrate concentration. So it's the density basically of that substrate molecule, or that molecule that is going to be acted upon by the enzyme, the substrate concentration at which the chemical reaction proceeds at 1/2 of its maximum possible velocity. So why does this exist? Why do we have this Km number, and why do we have this equation in general? I just defined all of the terms for you, and I'm going to go back and just run over them again, and then we're going to talk about why this equation exists, why we need to use it. Okay, so or no, let's start with substrate concentration. So substrate concentration, S in brackets, is a molarity or molality, or whichever concentration unit you want to use, but I think pretty sure it's molarity for like most of the time. The substrate concentration at which the reaction happens to be proceeding. The V naught or V0 is the speed at which the reaction is proceeding at the specified substrate concentration. And then the V max is the highest possible velocity of the reaction, or the highest possible rate at which the reaction could be occurring. And then the Km, or Michaelis constant, hiding over here, is the substrate concentration, so it's a molarity too, at which V0, or the rate of the reaction, so I'm going to say V0 here, V0 is 1/2 of Vmax. Okay. So the rate of the reaction, the substrate concentration at which the rate of the enzyme's reaction is 1/2 of the maximum possible rate. So why is this important? Well, this equation allows us to plug in random numbers for substrate concentration and find out how fast the reaction is going to proceed. And that could be really important if we're trying to determine how fast an enzyme catalyzes the reaction that it is catalyzing. The purpose of having enzymes in our body is to make reactions faster. And the importance of the Michaelis-Menten equation is to tell us how fast. So it's really quantifying the most important part of the enzyme's job. It's quantifying how fast it is making its reaction. So that's that V naught. Okay. What are the sort of variables that we can play around with? Well, it's really substrate concentration. If substrate concentration is high, you would expect V naught to be higher. If substrate concentration is low, you would expect V naught to be lower. That's because the concentration of chemicals determines how likely they are to sort of collide with each other and catalyze the reactions, or and produce the products that they form together. So that's why substrate concentration is so important. V max is important because it's the highest theoretical rate at which the reaction could be going, and different enzymes are going to have different V maxes. An enzyme with a higher V max is going to be faster. An enzyme with a lower V max is going to be slower at its maximum possible velocity. This by definition. And then the Michaelis constant. So if the substrate concentration at which the reaction velocity of half of its Vmax is really high, okay, so let's say high Km. All right, I did Control M instead of Km, so I have to find it again. Km, high Km equals, let's just say worse enzyme. Low Km equals better enzyme. So often on the MCAT, you have like up, you have up and down questions where the question is essentially, well, this thing goes up. What happens to this other thing? Does it go up or does it go down? And then the answer is completely based off of whether or not you understand the concepts that will lead you to say whether it goes up or down. This is an example of this where you would think, oh, bigger number, faster enzyme, better enzyme. Not always. Especially when you have a number like Km that's here in the denominator. That's going to actually make the enzyme slower, aka worse, because the whole point of the enzyme is to go faster. I'm just going to call it worse. It's sort of like a quick and dirty way to understand it. Okay, so high Km is a worse enzyme because it is faster at a really high S, and that means it takes a lot of substrate, i.e. a higher substrate concentration, to get going. And if it's a low Km, it takes only a little substrate to get going. So the Michaelis constant is telling us how effective this enzyme is at doing its job at the present moment. How good is this enzyme at doing its job? And that becomes really important when we talk about regulation, because what an inhibitor is going to be changing is something like the Michaelis constant, which determines how fast the enzyme is doing its job. And the purpose of the regulator is to inhibit or increase the enzyme activity. And this is one of the mechanisms by which it does that. Cooperativity, we're not going to talk about that. And right now, feedback, I just want to hit the important topics, especially the ones that are related to KM first, because we just talked about KM. Okay, let's talk about the types of inhibition. Okay, so on the MCAT, you're going to encounter inhibitors. So we've talked about enzymes as sort of like these managers that are seeing these potential reagents, and they're saying, okay, time to get together right now, do your job right now. Time's a wasting. We're thermodynamically favorable, but why don't we just do all of that faster? But who's managing the managers? It's almost like a structure, like a corporate structure. So who's sitting above the enzymes and sort of like managing how fast they're doing their job? That's these inhibitors. So an inhibitor could come from within the body, which we're going to see examples of here, or it could come from outside of the body in the form of a drug. So a pill, such as an ACE or acetylcholinesterase inhibitor, I think that's what it stands for, is like a kind of high blood pressure medication. And inhibitor means that it's inhibiting the acetylcholinesterase. And we said before how the ACE is the general ending for enzymes. So an enzyme inhibitor isn't necessarily something that comes from within your body. It could be a drug. And often that's the purpose of a drug, is to inhibit a certain enzyme that might be in the body. Okay. So let's look at these different kinds of inhibitors and the different effects that they have on all of these variables that we talked about before when we were quantifying the activity of enzymes. So let's look specifically on the effect that they have on Vmax, or how fast the enzyme can work, and Km, aka the substrate concentration needed in order for the enzyme to reach a half of its maximum velocity. Okay, so we're going to define the inhibition types by the effects on via its effects on, and then we're going to do 1 is Vmax, and then 2 Km. Okay, and it's going to be either up or down. So the MCAT is all a test about ups and downs. So that's what we're going to be talking about here. Okay, awesome. So this, again, is the Miles Down MCAT review sheet. It's very sort of quick and dirty, but it gives a really good general picture of what you need to know for the MCAT. So let's start with competitive inhibition. A competitive inhibitor does not change Vmax, but it increases the Km. So going back to what we talked about, a high Km, worse enzyme. That's how it's an inhibitor, is it makes it a worse enzyme. How does it make it a worse enzyme? By increasing the Km. So Vmax is unchanged, and Km and I remember hearing a mnemonic for this once that was something that along the lines of like KM K climbs with competitive. And that mnemonic actually accounts for the entire activity of the competitive inhibitor for the purposes of the MCAT in the way that we're simplifying it. Because if KM climbs and that's the whole mnemonic, that means that nothing happens to Vmax. So KM climbs with competitive. I think that's called alliteration, where you have like the same sort of syllable at the beginning or the same like sound. So KM climbs with competitive. That's a good way to remember competitive inhibitors of enzymes. That's the first type. And the mechanism, which I think is actually less important to memorize, but still, it's almost as important actually. The competitive inhibitor binds at the active site. So it competes for the active site of the enzyme, aka the lock component of the enzyme, competes with the substrate for the active site, aka the lock of the enzyme. Okay, and then we have non-competitive. Okay, so we did the competitive ones. Now we're doing the non-competitive ones. So these names are purposefully confusing. There's probably better ways to describe these different inhibitors, but we're using these names because archaic names are part of what the what the MCAT is testing you on, is whether or not you can recognize an archaic name and make a connection to something useful and important and relevant in real time. Again, remember the doctor connection. It helps explain a lot of this seemingly random stuff we need to memorize. Okay, non-competitive inhibition is when the inhibitor binds with equal affinity to the enzyme and the enzyme substrate complex. We can talk about that later, but first let's see about Vmax and Km. Vmax goes down and Km is unchanged. Okay, I'll go back to my document and say that Vmax down and Km is unchanged. Okay, so non-competitive decreases the Vmax, i.e. the fastest rate at which the enzyme could be acting, and Km is not changed. So the substrate concentration at which the reaction proceeds at half of its maximum possible velocity is not changed in a non-competitive scenario. So with a non-competitive inhibitor added to the enzyme. And why is this? This is because the inhibitor binds with equal affinity to the enzyme and to the enzyme substrate complex. I'm just going to drop that entire sentence into here because that is kind of important. So the inhibitor, which is either the drug or the molecule that comes from inside the body or the compound that comes from inside the body, it will bind with the same affinity to both the enzyme or the catalyst that's catalyzing the reaction and the enzyme substrate complex. That means that the highest possible speed that the enzyme will be able to do its job at is reduced because the enzyme and the enzyme substrate complex are being bound with equal affinity. It's in both places at the same time. So that's decreasing the maximum possible rate for this reaction to proceed, but it is not decreasing the amount of substrate it takes to get to that lower Vmax. So that's non-competitive. I don't have a good mnemonic for that one, but I know it's not competitive, which competitive competes and Km climbs. So Km increases. Here, Km is unchanged and Vmax goes down because we're binding with equal affinity to the enzyme and the enzyme substrate complex. I'm not giving a full throat definition or explanation of why that is. Here. Uncompetitive. So we've talked about one where Vmax is unchanged. Vmax goes down. Km increases. Km goes down. What about one where Vmax and Km both go down? So that's sort of like a bad thing and a good thing happening, because as we've discussed with Km, the directionality is flipped. It's higher Km is worse, lower Km is better. So what if they both go down? And that is uncompetitive. And that is because the inhibitor is binding only ES, or the enzyme substrate complex. Okay. And then mixed is, we're going to say for now, it depends. What you need to memorize is these definitions. Competitive, Vmax is unchanged, Km increases. Non-competitive, Vmax goes down, Km is unchanged. Uncompetitive, both of them go down. I sort of like to think of this in the let's see, in the context of medical school applications. So we often talk about a competitive versus an uncompetitive applicant. And so I think of like everything is going down if you're an uncompetitive applicant. And that's how I remember that Vmax and KM both went down. And then I try to just put non-competitive and just out of my brain. These are purposefully confusing. Again, the MCAT is testing us on archaic terms that are no longer really used. These might be used, but generally they do that. So uncompetitive, you're an uncompetitive applicant, everything's going down. Competitive, KM climbs. Non-competitive, it's the other one where KM is unchanged and Vmax goes down. Vmax has to go down because it's an inhibitor and KM is unchanged. So something needs to be changing to make it slower. Okay. So we've covered these types of inhibition, and now let's just go through these last few points that the AAMC outlines. So regulatory enzymes. Okay. So let's go to the miles down guide and look at what they say for regulatory enzymes, specifically allosteric enzymes. So an allosteric effector binds at the allosteric site and induces a change in the conformation of the enzyme, so the substrate can no longer bind to the active site. Okay. So I'm just going to Google what they mean by allosteric enzyme, because this is saying allosteric effector. Okay. Okay, great. So by allosteric enzymes, I guess the AAMC is like reducing an allosteric effector to an enzyme and saying we just want to test students on the enzymes that might bind to an allosteric site and induce a change in the conformation of an enzyme. I'm not really sure, but generally allosteric means not at the most important steric position or three-dimensional position in a molecule. So an allosteric effector is going to be binding at what's called the allosteric site. So notice that when we were talking about sites before, we were talking about the active site, aka the lock of the enzyme in the lock and key analogy. So if we're binding at something that is not the active site and is instead the allosteric site, then we're binding at a different place than the substrate binds, because the substrate binds the active site. Okay, so the allosteric effector binds the allosteric site and induces a change in the conformation of the enzyme, so that substrate can no longer bind to the active site. So it's almost like changing the active site from afar. It's binding at this other place and inducing a conformational chain reaction in the three-dimensional structure of the molecule, such that the active site no longer binds the substrate. It's really important to note, because it's a tricky kind of concept. You would think that you need to jam something into the lock in order for it to stop working. But what the allosteric enzyme does is it just kind of pushes the whole door into the frame so that you can't really open it anyway, no matter how hard you try to get to the lock. It obscures the lock, if you will. It doesn't put itself into the lock. Okay, to use the lock and key analogy. So allosteric enzymes okay, this is a really awful sentence. Allosteric effectors bind a site other than the active site, called the allosteric site, and block the active site from afar. That's Aiden's definition. That's not like, that's not the definition that you would find in the textbooks I cited above. And then let's see, covalently modified enzymes. These are pretty self-explanatory. Yeah, enzymes whose catalysis involves the formation / breaking of a covalent bond. Or maybe they're trying to get at enzymes that are modified the formation breaking of bond. Honestly, I'm not sure. And then, azimogen. Okay. I'm not sure if I'm pronouncing this correctly, but the zimogen or zymogen is like the pre-enzyme. So often in the body, we're trying to get a molecule from one place to another before we want that molecule to do its job. And one way to do that is to have a precursor to that molecule secreted from one place and then sort of changed into its active form only where it's needed. So it's not doing its job throughout the whole body. It's only doing its job exactly where you want it to. One way to do that is via the zymogen pre-enzyme. And I think the way I remembered this is, I remember, let's see, the letter Z is like the end product of activation. So again, this is like totally just coming from me, my memory trick. It's the letter Z. You don't usually have a term that starts with the letter Z, but the letter Z is at the end of the alphabet, and it is azimogen is the precursor to the end product of the enzyme activation. That's the way I remembered it. It's a really sort of roundabout memory trick. Feel free to use it, but it's okay if you don't, because it doesn't make a lot of sense. But I find that it's actually the more sort of quirky mnemonics that you remember for yourself but don't make a lot of sense that end up actually sticking and helping you on test day. Okay. So again, it's really roundabout, but that's what we're doing. I want to go back now and just see if there are any terms, and then I'm going to do a quick recap of everything that we talked about today. Okay, feedback regulation. And then we're going to do a question. Feedback regulation is another way that enzymes are regulated, and it is by the products of the reaction that they catalyze. So I'm going to say that when an enzyme catalyzed reaction the enzyme slows down. This is a very simple way of putting it, but this is one of the ways that our body keeps our chemical reactions in check. We don't want too much of a good thing. We don't want our enzymes, or sorry, the reactions that those enzymes catalyze to happen ad infinitum, or else we would be poisoned by the products and our cells would be overwhelmed by the products. So, one very simple way that nature has of helping us avoid that sort of predicament by having feedback regulation, where the products themselves slow down the enzyme just by nature of their existence. And there are going to be examples of that on the MCAT, one of which I hope to share in this live stream after I finish with these terms. Um, Cooperativity. Um, let me. So I'm going to define cooperativity sort of based off of what I remember now. But again, consult one of those resources above for a more full, thorough definition. When the binding of one substrate to an enzyme / protein influences the likelihood of that enzyme / protein to bind another substrate. And the classic example is the oxygen carrying molecule hemoglobin. It turns out that our body's way of knowing to take oxygen from the lungs and put it into the tissues is not knowledge in the human sense, but it's by using cooperativity, which means that it takes the oxygen, it takes one molecule of oxygen that makes it more likely to take another, and then another, and then another. That's happening in the lungs, and then essentially when it gets to the tissues, it starts dropping off one, and then the next, and then the next, and then it becomes more likely to bind CO2 in the tissues, and then it drops it off in the lungs. So it's sort of that cycle. It's not knowledge, it's cooperativity. It's a property of the structure of the enzyme or the protein in the case of hemoglobin, the oxygen carrying protein. Okay, and then let's see if there's anything else that we talked about. Mechanisms of catalysis. Let's see. I'm going to skip that for now. And then let's do an MCAT question. So now I'm going to share my screen from the AAMC. I'm going to log in. That I'm not going to show on camera. But let's see. All right. I'm doing two-factor authentication for the AAMC login. So now what I'm doing is I'm going to the the Association of American Medical Colleges. I'm logging into my own MCAT test prep hub, and I'm pulling up practice exam 3, which rounds out its biochem section with a question about feedback regulation, if I remember correctly. So let's go to that test. I'm going to share my screen now. So this taking of it, I did it back in March of 2025, 522. I mean, it goes to show that your score can. Okay. I think it's question 57 that we're looking for. Okay, perfect. Okay. This is what we call a discrete question on the MCAT, which means that it is basically like a gun to your head question. Know it or you don't. Answer it now or you don't know it. Whereas other questions will give you a whole paragraph before you are presented with the questions. This one is just a question with a tiny little blurb, and it's testing you on a very small amount of knowledge that you either have or you don't, which is why it's so important to memorize. That's part of what we're doing today. Okay, in the reaction below, the activity of glutamine synthetase. So ACE sounds like an enzyme, and we're talking about the activity of glutamine synthetase. So probably something that's synthesizing glutamine decreases as the concentration of tryptophan increases. Okay, so glutamine and tryptophan, well, that rings a bell, amino acids. And then synthetase is going to be, glutamine synthetase is going to be our enzyme or the protein-based catalyst that acts upon glutamate to make glutamine. The number of glutamine synthetase, so the number of enzyme molecules in the cell, remains constant regardless of the tryptophan concentration. These observations are consistent with a mechanism of. Okay. So, the number of glutamine synthetase, this is the first time I've seen this question in a while. So, we're going to answer it in real time, which is very good prep. The number of glutamine synthetase molecules, so how much enzyme is in the cell, is constant. It doesn't matter how many. It doesn't matter what is happening in the cell. The number of enzyme molecules, I guess, is constant. These observations are consistent with. So, which mechanism that we've learned for controlling the activity of an enzyme can occur when that enzyme is at a constant concentration in the cell? Okay. Well, tryptophan is okay, so tryptophan is the end product of this reaction. We've learned that when tryptophan increases, so you get more product, the activity of glutamine synthetase decreases. So activity and number of glutamine synthetase molecules are two different things. You can have enzymes that are not acting at their best because of inhibitors, as we've learned by the competitive, uncompetitive, and non-competitive inhibitors we discussed before. So here, there must be some other way that less tryptophan is being produced. It's not that we're getting rid of glutamine synthetase, the enzyme, it's that we're inhibiting its activity. Okay, so we know that inhibition is going on. Let's look at these different mechanisms. A, translational control. Okay, so controlling glutamine synthetase at the level of translation. This doesn't seem likely to me because the number of glutamine synthetase molecules is not changing. And this isn't like a translation transcription sort of lesson, but with translation, you're connecting the mechanism of inhibition to the number of end product proteins that you make. So we've just been told that the number of those proteins doesn't change. So I'm going to say it's not translational control. B, glutamine degradation. That doesn't really make sense to me. So I'm going to skip it for now. C, transcriptional control. I would say the same thing with A here. We have the same number of glutamine synthetase molecules, so I doubt that the change is occurring on the level of transcription or translation. And then we have feedback inhibition. So we just did a lesson where feedback inhibition was flagged as this really commonly tested mechanism of enzyme regulation. So we're sort of primed to think about that, but let's think deeper as to like why this is clearly feedback inhibition. How does feedback inhibition work? Feedback inhibition is such that the enzyme concentration doesn't change, but the enzyme's activity goes down. That's the inhibition. Um, so the question is asking whether the observation is consistent with feedback inhibition, because that's one of the answer choices. So the number of glutamine synthetase molecules doesn't change. Sure, it doesn't change when there's feedback inhibition. But as tryptophan increases, the activity of glutamine synthetase decreases. So what's happening here is that the product is going back and inhibiting the enzyme. It's not getting rid of the enzyme, it's not changing the enzyme in number, but it is stopping it from doing its job because there's already enough tryptophan. And if you had too much tryptophan